growth and what this means. So we've been running things in lab. There are nutritional factors. So we have major elements, trace elements, and growth factors. And all of these affect how our colonies are going to grow. We also have physical factors like temperature, pH, oxygen levels, osmotic pressure. These are also things we need to consider that are going to affect the growth of our organisms. So, what is growth? What is microbial growth? What does that mean to us?
The number of bacteria or organisms.
Number of bacteria or organisms, okay? So, simply it's increasing microbes. It's a lot of times what we actually end up looking at and wanting is to see the growth of colonies, which means a single colony means it came from a single cell. So it's that one cell that's multiplied into a number that we can see in a colony. So those are discrete colonies. So, and with these nutritional factors, there are a number of different things that cells need, and they acquire them from multiple sources, which we'll talk about a little more. Okay, so major requirements. Everything needs carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, the most common elements in life. I mean, can anyone tell me why? Why, where do we see these? Why are these so important? Where have you seen them?
They're in the air.
Well, yes, oxygen's in the air, we breathe, but think about the DNA. What are the, we'll talk a little more later about, the actual elements that make up DNA, that make up the proteins. They all come from these basic elements, okay? So, we are organic we are organics. We come from carbon. That just means it has carbon in it. These are just the most common elements. We need the components to make proteins, make carbohydrates, lipids, nucleic acids, DNA. All of these are just important major elements. Trace elements, so we need just a little tiny bit of these. And these are things like copper or cobalt or zinc or magnesium. So these are things that the cells need to grow, but you don't need them in nearly the quantities you need to have carbon. Carbon's kind of essential. Growth factors. So, what are the organic compounds needed that cannot be synthesized by microbes? So this includes some amino acids or vitamins. We talk about there are 20 amino acids, and some of those we actually can't synthesize for ourselves, so we have to eat something to get those essential amino acids. And so that's one of those growth factors of things we cannot make for ourselves. Microbes also have some things that they cannot make for themselves that they must acquire from somewhere else. Okay. So, energy source, carbon source. Very important little things. We have photo. So that means your energy source is light. You see the photo. Versus the chemo, which is where it's chemical compound, so organic molecules. Autotroph means you're doing CO2, which means that you're being able to make your own food source, versus heterotroph, you're needing to take in a food source, an energy source, okay? So chemoautotroph means that you need chemical compounds for your energy source, but you can produce your own food. Or a chemoheterotroph, you need chemical compounds and you need organic compounds for your carbon source. Okay? Carbon sources are incredibly important, and it's a lot of how you can change and manipulate organisms. So autotrophs are also a lot of times, they're known as the producers. They're usually bottom of the food chain. They're the ones that are making the energy for the rest of the chain. You can heterotrophs. They depend upon energy and carbon fixed by other organisms a lot of times. So these are the consumers, or also the decomposers of your organism. Okay. So, pretty little quick picture to separate what we just saw with what is your energy source, what is their carbon source, and just to also give you some examples of some of the organisms. So plants, algae, cyanobacteria, photoautotrophs. They can turn light into food. And then you have your green non-sulfur bacteria and purple non-sulfur bacteria, being your photoheterotrophs, aerobic respiration. So most animals, fungi, we're in here, the chemoheterotrophs. We kind of need to consume food for energy and as our carbon source. Chemoautotrophs. So the nitrifying bacteria and some archaea fall into this category. Is this making sense? Questions? I see a question. Question? So what does this all mean? We're always looking for what is our optimal growth rate. And I can tell you this is something that we actually still do in lab a lot, because every time you've mutated a strain, you have to see where is its optimal growth. And one example here, you can see, looks like bacteria, but if you're looking at a different temperature scale, based on the temperature, you can have it cold, 22 degrees, and that means everything in that metabolism is going to slow down, and it's going to grow very slowly. Whereas if you ramp it up, everything's going to speed up, you'll get faster growth, but you might also cause problems because it's not used to that high of an extreme. You might even kill it if you go too high. Then you got to try and find that optimal spot, that sweet zone, where it is really producing the most, the highest level of growth. You have your growth rate, and then what temperature gives you that optimal growth rate. Temperature is important. So that optimal growth temperature is important. Not just because we want it to grow as quickly as possible, but also there are a lot of effects that can happen based on temperature. If the temperature is too low, then membranes can become rigid, fragile. If the temperature is too high, membranes can become too fluid. So you can see how you have this window where you really want the temperature to be. And it all depends on the different organisms. So we've got 4 different ones we're looking at. The psychrophiles, their optimum temperature is below 15 degrees Celsius. So what is room temp about usually?
22.
Celsius. Talking Celsius, we're in science. About 22, 25, it depends on how cold they like to keep it. So below room temp. You can also then get the mesophile. Their optimum temperature is between 20 and 40 degrees. What temperature are we at? Celsius. We're at about 37. Sorry, I said 97. 37 degrees. Okay, so that's right where we're at. Yeast, bacteria, coli, they really like this pretty zone. Thermophiles, they like 45 degrees Celsius, so that's pretty warm. We're talking hundreds in Fahrenheit. You don't want to go outside on days like that. And then you have the hyperthermophiles, which are in the extreme temperatures. So these are a lot of times, you have these like the archaea, in those thermal vents, in just some of these really weird random places. I think the highest they have found so far is they found one that could live in 120 degrees Celsius. So what's boiling in Celsius?
100.
100. So 20 degrees Celsius above boiling is rather impressive. So just to kind of look at what we're talking about when we talk about these, the temperature ranges. And so for each of them, they have their own little curves of where their optimal growth is. What is their optimal range? Okay, for that broad category. And so if you're ever doing experiments with them or you're ever wanting to test them, usually you just go for the middle of this and then you can tweak it left or right based on the exact individual organism you're looking at. Okay. And then just because it's weird that anything would want to grow on ice, a little picture. Because, I don't know, to me that just seems weird and ridiculous because it's incredibly cold. But there are organisms that can grow at really low temperatures. Another thing that's very important for microorganisms is pH. If we play with, if you ever put bleach on your hand and you feel your hands get all slippery, that's because it's a base and it's kind of eating away. And so one of those things where we have certain ranges we like. We are neutrophiles. We prefer a neutral type pH. So anywhere between 6.5 and 7.5, 7 being neutral. Most bacteria are neutrophils. You can get the acidophiles, and some bacteria do this. Many fungus can deal with this, and they can get as low as pH 0. So, I mean, we're talking probably, was it our stomach acids at pH 2 or something like that? So, it's pretty impressive what they can survive. And then you also have your alkaline files. So, acid or alkaline, which side of the pH scale are you on? And so they go up to 11.5. So, I think bleach is only at 10-ish, maybe even only 8, because it doesn't burn that much. So it starts to give you an idea of how extreme these can be. Now, can anyone think why would you really care about the pH levels besides that? We know it burns us, but why would they care, or what chemical reaction is happening that's making this matter? What is pH?
It's a scale of acid to
Okay, so pH, we're talking about, how free are those hydrogen ions. So with the, with pH, we're talking about the hydrogen ions, the hydroxyl ions, and if you have a lot of either hydrogen or hydroxyl ions, so either acidic or basic conditions, that's going to mess with hydrogen bonding. How are you going to form new compounds if you can't do basic hydrogen bonding? How are you going to have stable interactions if you can't have that hydrogen bonding? So it can really start to affect how that organism survives. Does that make sense? Oxygen. We all know and love oxygen. It's required for us, but oxygen can be incredibly hazardous. I mean, we've all heard of the free radicals and all the latest health craze. Get rid of all those free radicals. But there are toxic forms of oxygen, and one of the things is oxygen is highly reactive. It wants to react. And so that means you can get, the superoxidized radicals. And so those want to go and they want to be balanced. They want to find those protons, and then that means they're taking that out of the system. Or that means that a lot of times they can actually take it, maybe pull it off a piece of DNA, and so now all of a sudden you have damage to your DNA. You can have superoxide radicals, the superoxide dismutase, you need catalases, you have peroxidase. I remember I had one guy who, way back when I was in community college, saying, look, I have this super oxygenated water. And you're like, you're drinking oxygenated water. That's hydrogen peroxide. Your body knows how to handle that, but why would you intentionally put extra work? So, we have ways of handling them. We have multiple repair pathways to deal with it because oxygen can be incredibly damaging. Makes sense? Free radicals, hydroxyl radicals. Oxygen likes to react and create problems. Okay. So not only can oxygen be hazardous, but it is required. So 3 basic types: obligate, anaerobes. So grow only when oxygen is present. This would be like us, but because if oxygen isn't present, we don't grow very well. Facilitated anaerobes. So grow with or without oxygen, but they grow best when oxygen is present. So this is like yeast. So yeast will grow best when there is oxygen, and they will double like crazy. Starve them of oxygen, they'll start doing fermentation. So they will survive, they will reproduce, they will grow, not as well as with oxygen, but they will still survive. So facilitate. Or obligate. There are some organisms out there that cannot handle oxygen at all. And if they come in contact with the oxygen, they die. Just because they don't have the capabilities to deal with it. There are some aerotolerant anaerobes, which can tolerate oxygen because they have some kind of forms or enzymes that can actually detoxify the oxygen, but they really prefer no oxygen. Or the aerophiles, which want just a little bit of oxygen. They don't want much, they just want very, very low levels to make them happy.
What were those ones called?
And this was the one that comes with me. So, aerotolerant anaerobes, and then the aerophiles. There's too many places to go with that word. Okay, so, temperature is important, pH is important, oxygen levels are important. Nitrogen, one of the other most common elements, is also incredibly important. Okay, so nitrogen can be a growth-limiting nutrient for many microorganisms. With yeast, in a diploid format, if you leave them alone, they're going to keep producing diploids and doubling. If you starve them, don't give them nitrogen, then they're going to go and start making tetrads because they're going to think life sucks, we need to mate and recombine and try and get some variance going. So if you don't have nitrogen, it's a growth-limiting nutrient. It can be acquired through organic or inorganic nutrients. So organic just means what?
With carbon.
With carbon, okay. So one of the things that's kind of interesting is that nitrogen is, 79% of our atmosphere. But the problem is that it's N2. So remember, we talked about this, is that you have that nitrogen, triple bonded to nitrogen. So we have a lot of nitrogen in the atmosphere. But most organisms cannot utilize that form of nitrogen gas. So this is why we need the nitrogen fixers, which some of the bacteria do, to turn it into ammonia so that we can actually access that nitrogen. Okay? So, nitrogen gas, tons of it, free gas, free nitrogen throughout the atmosphere, but not in a usable form. Question? And the last thing we were talking about was pressure, osmotic pressure. So we talked about hypotonic, hypertonic, depending on the amount of solutes. You also can have pressure, osmotic pressure based on depth. And so how deep you are in an ocean. So organisms need water to dissolve. Their enzymes, their nutrients, pretty much life is dissolved in water. And so if you're not being able to regulate that water, you start to have some issues. So we have osmotic loss from a cell. So if you start to lose water from a cell, you need to try to balance that osmotic pressure. And so some organisms have actually figured this out. If they want to live in high salt conditions, they start turning themselves into haplophiles, salt-loving organisms. So they start having more solutes on the inside, so that way they can balance out how many solutes are on the outside. And so you kind of end up getting, they figure out how to balance that pressure so that they aren't losing all of their valuable water. So you can have haplophiles, salt-loving organisms, osmotolerant, so they're facilitated haplophiles. They're able to tolerate, so anytime you're seeing that facilitated, which means they're able to tolerate it. Also, you can have barophiles, which means that they can range in extreme pressure, so they can start being underneath in the ocean, different levels, because different depths in the ocean give you different water pressures.
Can you spell that one? Baro, B-A-R-B-A.
Barophiles.
That's not right.
Water pressure is a proportion of depth. And I mean, they have to make sure that all of their enzymes and everything is good under those conditions. Because you can imagine if you get too much pressure, pre-flooded, whatever you can, if your proteins aren't stable, then the rest of your cellular life isn't going to be very stable as well. Make sense? Biofilms, quorum sensing. Okay, so quorum sensing is a process by which bacteria respond to changes in microbial density. They use chemical signals and receptors. They carry out this kind of sensing, going, okay, what is around me? How much is around me? So high concentrations of bacteria produce high chemical signals, basically saying there's a lot of us around. And then these high signals will trigger the bacteria to actually form into these biofilms. Okay, there's enough of us. Let's form this biofilm. Let's put the energy into the extracellular matrix that's going on. And so you start to actually create that entire biofilm. It starts, there's enough of them, they go, let's stick together. Let's actually start helping each other. And when you get levels like that, some of them are actually more dangerous as a biofilm than individually, just because of the sheer numbers and because they start sticking together and causing other issues. So, a picture of a biofilm plaque on human tube, just to kind of show you what it can look like with all the different organisms. Okay, so we're going to switch gears a little bit to how do these start up? Bacteria colonies. So a colony is a cluster of identical cells. Okay, so you have one cell, it divides. Those two cells divide. Those four cells divide. And so you can get exponential growth because it's doubling every single time. And so for the most part, you can get a lot of growth really quick. That's why when you guys did your open air plates, you can put them in the incubator and overnight you had colonies. The nice thing is when you look at discrete colonies, so a colony that is all by itself, you know that they are all clones of each other, that they all arose from a single colony. If there were two cells next to each other, or, you know, you would have seen, you know, or had grown close, you would have seen the colonies merge. But if you see only one colony, usually, you know, and we'll talk about doing streaking later, you'll get a single colony, a single cell. So an example is, it had a nice little picture of growing an E. coli colony. So when you streak anything, so when we streak something, you actually, microbiology, you're going to want us to do, you're doing a dilution streak is what you're doing. So you streak it this way first, and then you bring it down, and so with every pass, you are thinning out or diluting out the number of bacteria on your tip of, you know, your flame loop or the pointer thing. So that way, in the beginning, you're going to have a ton, and you can't tell the difference between those colonies. But by the time you get here, you have nice individual colonies. This is how you can obtain a pure culture, because if you, and we're going to be playing with mixed cultures today, you're going to then carry it through tomorrow and stuff, but you'll be looking at our, as you streak them out, at first, you're going to see the two cultures. But as you move through the plate and the dilution, but as you're streaking, you're going to start seeing the two different cultures, and you're going to see different colonies, and they'll be distinct, so you can actually separate and get a pure culture. Does this make sense? So, how do you make a pure culture? So, one of the most common ways to make a pure culture is on a Petri plate. So, you have your plate. In this one, they had red algae, which is why it's red. So you have polysaccharide, you have sugar source, carbon source, and you will, with a flame loop, you basically dip it into a liquid media. You streak it one way. And then you cross it a little bit for 2, you cross it for 3, and then you cross it for 4. And the idea is that you're dragging a little bit from, most of your stuff should be gone up here, but you're dragging a little bit down into the next set of streaks. You're dragging just this, whatever dilution you have here, you're dragging that into the next section of your plate so that you have a dilution factor. So this is just a streak plate method. You dilute the bacteria by spreading them on the agar plates to get the individual distinct colonies. The pour plate method, you mix your sample with some melted agar and you pour it into a plate. And so in this one, you're actually doing a 10-fold dilution multiple times. So 1 ml of your initial sample into 9 ml of broth, 10-fold dilution. Do it again, now it's a 100-fold dilution. Do it again, you get a 1,000-fold dilution. And then if you plate that again and do another one, then, you know, so you can start getting, you'll get, you know, at, say, was it 1,000-fold, you get a bunch of colonies, and then at one more 10-fold dilution, you'll get even less colonies. So with each dilution, you get less colonies just because you're transferring more over each time. 2 different methods make sense. You either just straight dilute it in culture, or you dilute it on the actual agar plate. Okay. Growth medium. So we talked about these agar plates. We talked about, you know, growth medium kind of thing. So